Thanks very much for joining us today for another one of our TESS seminars. TESS, of course, is our Center for Tropical, Environmental, and Sustainability Science here at JCU, which is one of our main vehicles for promoting terrestrial and aquatic research, but not marine research. It's an absolute pleasure to have Matt Luskin joining us. Matt was here about 4 years ago, but I don't think we probably have any overlap at all, except for a few of us, in terms of seeing Matt's talks. Matt, a lot of things you could say about Matt. Matt was trained in the US. He got his undergraduate degree at UCLA, one of the world's top, if not the top public university, vying along with Berkeley, where he got his PhD, which is the other top public university. But anyway, he's had fantastic training. He spent a lot of time in Southeast Asia, been affiliated with the Smithsonian, with the National University of Singapore, and with other institutions there. Matt has really carved out a good niche for himself here in Australia as founding and leading what I think is called the Wildlife Observatory of Australia. And so he's really establishing himself, or has established himself, as sort of the camera trapping guru and the person that you go to when you have these, complex problems. And he's dealing with very thorny real world data sets and harvesting a tremendous amount of information. Matt is an associate professor at the University of Queensland. It's great to have him here, and we also see him when we go down to Brisbane. But anyway, without further ado, we'll thank Matt for joining us for Cameras for Global Change Ecology. Matthew.
Thank you, Bill. Before we get started, just want to acknowledge the traditional owners of the land we're on here, and also where I work in Brisbane. And I also want to thank Bill and give him a huge congratulations. He was elected as a Royal Society Fellow. It's a very prestigious honor. And I think my long relationship with Bill, I can really say that the statement made by the president is really accurate for Bill's career, which is that his achievements represent the very best of scientific endeavor, from basic discovery, research with real world impact across health, technology, and policy. He's one of the few people who I know has done that. And he's also been so welcoming. People a lot of time when they get a little bit further on in the career, they have less time for people who are coming up and maybe being dismissed. And Bill has always taken the time to have a conversation with me and to guide me. So congratulations to Bill. I'm going to talk today about a variety of things, but one concept that I'm going to talk about a lot is ecological cascades. You may have heard of these often just as trophic cascades, but I'm going to talk about them in a little broader context. And then I'm going to focus a lot on how we use cameras to study ecological cascades at large scales and across ecological gradients. So I want you to focus on the one on the left with the hunter, where I explain, or this image will explain that when there's a defaunation cascade, hunters often target all the large animals. And so you can get a release of small animals, and that has huge impacts for the tree community because the small mammals are often seed predators killing seeds as opposed to larger animals that are seed dispersers. The most famous cascade is a trophic cascade from the loss of apex predators, which releases prey, and they often become abundant and they overconsume the vegetation. You've heard of the wolves of Yellowstone example. And one that I'm really interested in, and I think has been underappreciated to date, is a bottom-up food subsidy cascade. And what this refers to is an increase in the frequency of forest producing fruit, or if we're in a grassland, really good, or in a desert, really good rainy years that produce lots of vegetation and the herbivore populations boom. Or it can be due to a human effect. It could be crop raiding. This is an image showing oil palm plantations, which I'll talk about more, but it could be up in the wet tropics, animals eating sugarcane or eating fruit nearby people's houses. And so that can create more food for the herbivores, which then could cascade up and lead to more predators. So each of these big issues comes along with a management strategy that is dominant. So if hunting is the key issue, a lot of time you'll see protected areas and enforcement of hunting, especially in protected areas. If you have trophic cascades, a lot of time you have carnivore conservation or reintroductions. And if there's problems associated with food subsidy cascades, you might see, or an increase in the natural food cycles. Those could be addressed through climate policy or land use policy. And as simple as those three things are, the truth is it's actually a lot more complicated. All these things are interacting with natural processes, ecosystem dynamics, and those all fit in together to make for quite a complicated story. And I think that complication is really the exciting part and what makes ecology fun. We will never figure out everything, and every site is unique and has new things to learn. But the premise of all these arrows is the more arrows you add, the more data you need. And that is especially true in messy systems, and ecological systems are among the messiest within science. And so what this means is we need huge data sets. So how do we track and study these complex ecosystems, especially when they're changing rapidly? So the method that I have been really leaning into is the use of camera traps. These are simple, cheap pieces of technology that can be strapped to a tree, left for 2 to 6 months, sometimes longer, if you're out in an area with sunlight and they can have solar panels. They take images they see in heat, like infrared, and so whatever walks by that has a warmer body temperature than the background, it would take a photo. So if leaves are moving, you usually don't get photos. So really easy to set, efficient, and produces a lot of information, and most importantly, extremely uninvasive for the wildlife there. Not capturing animals, not inducing stress, not changing how they behave in general. And if many people are setting many cameras over many years at many sites, we can get to a place of big data. And by big data, I don't mean, oh, we have 100 observations or 1,000 observations or 10,000 observations. I'm really talking about 1 million, 10 million and more observations, and many, many, many more absences, which is a very good data point, which means the camera was there, was looking, the animal was not detected. So it's not just the number of detections, but I will report a lot of these things in detections. Let me give you an example of why I'm going to build up to why we need to be working together and have these collaborative approaches, which I'm going to refer to as an observatory approach, is because I learned firsthand how long, hard, expensive it is to set cameras. So in my PhD, I spent 13 months out in Sumatra. I set more cameras than probably any other PhD student up to that time. I've been surpassed by my own PhD students and others. And I was trying to study tigers. And so I did this very large study, had 1 million photos. This is before we had computer vision to help us identify what was in the images. So luckily, there was a lot of enthusiastic undergraduates who sorted images with me. So most people come to the talks because they want to see cool pictures of animals. I am not going to disappoint you. I have lots of pretty cool images, and I'm going to let them speak for themselves. I'm going to start with some large carnivores. And I'm also going to start with a focus on Asia, build up into Australia, and then focus in on JCU and surrounding ecosystems right around here. So these are our 4 carnivores in Asia. And what's important to know is they're really rare. And so what that means is we have to sample an incredible amount to get enough data to make credible inferences about what their populations are doing. So 13 months, what did I get? 59 pictures of tigers, 59 unique tiger detections, of which there was 26 individuals. This was enough for one paper, but it was barely enough. And what was crazy is I had to go through 1 million photos to get there. And you're thinking, well, what are all those other photos? I think half of them were macaques. And so it is funny, it is cute, but this points to maybe hundreds and hundreds, if not thousands of hours of people's time going through images that are not going to be informing your research questions or are relatively benign, right? Just going through the same image over and over again of monkeys playing with cameras. Therefore, this is going to be a motivation for later on when I talk about the use of AI to help us process these things really quick. All right, so in 13 months, 12 people, 3 parks, I only sampled these things once. And realistically, to estimate what's happening with tiger populations at large scales, we need to do this all over the tiger range. And this is just for one species. And what this highlighted to me is we need to work together. We need to collaborate. We need to share data. I knew about the power of collaborating and sharing data and this observatory approach through my work at the Smithsonian. They have the Forest Global Earth Observatory, 100 plots all over the world spread from tropical regions to the north. These are vegetation plots with standardized methods where everyone can share data or at least request to share data. And the outcome of these plots has been over 1,000 research papers, I think over 60 papers in Science and Nature alone, of which Bill and others that we work with have been part of. So starting in Asia, we started putting together camera trap data sets. So to do this in an equitable way, we got everything together and we published a data paper. Anyone can go and access it. It has 239 camera trapping studies, right, all over the region, of which there are almost 500,000 detections. That includes both mammals and birds. This is showing genuses, not species. The species, I think, is 300, 350. And what that did is it catalyzed research. So there was over 20 papers in 2 years using this data set on a lot of these animals had never had a dedicated research paper before. And not just these niche species, niche meaning esoteric, but also high impact work. People really use this for some high impact work. One of the ways that my lab used it is we were interested in how the spread of oil palm plantations throughout the region was influencing animals and potentially even conflicts between humans and animals. And so what this graph is showing is we took that CamTrap Asia data set and we used the covariate at that landscape scale to say, what was that landscape like? Was it near, was the forest quality? So was it more logged near an edge, or was it pristine? And what was the situation with oil palm? Was there a lot of oil palm in that landscape or not? What we found is there was a set of general species, and those that work in the region won't find this too surprising, that really increase around oil palm and edges. And this has a huge ecological impact because these species crop raid, but they also are some of the key hosts of zoonotic diseases and really change the spread, the risk of disease spread between humans, wildlife, and livestock. Another way we use this data set is, this is work led by Jed Brody, is we got all the data and then we also used data from eBird, and we looked at whether or not the studies were conducted inside, nearby, or outside protected areas. And we didn't just look at the species diversity in terms of species richness. We went a little bit further and we looked at functional richness, which is what kind of ecological guilds and processes these species might perform, and also phylogenetic diversity, which is how unique they are evolutionarily. And what we found is for birds, protected areas did increase all types of diversity, which is a really strong result for birds. And it's really important because the UN has a goal of increasing the protected area coverage to 30%, which is a big increase. So we need evidence to support such huge policies. For mammals, it was a little more complicated. There wasn't a direct effect of a reserve of the site being protected. But for unprotected areas, there was a big effect of whether or not it was near a large park or near a small park. And what we think is underlying this result, the process underlying, is likely that mammals need big areas to sustain their populations. And so small parks are not going to be enough to maintain a lot of these mammals. Okay, so now let's move on from Asia. And let's just take a quick look at a small portion of the camera trapping studies globally. Okay? So we were able to do a lot with camera trapping studies just from Asia. And this is a fraction of the studies, only those that had the images stored and processed on a platform called Wildlife Insights, which is a great program that I use, although we're doing a new one in Australia that I will tell you more about, specific to our ecosystem. What this indicates is there's a lot of approach to scale up that we did in Asia across the world. That is especially true in Australia for a number of reasons. But so far, work in Australia with cameras and often in other ecological spheres has been relatively siloed into specific labs or specific government agency or NGO, less collaboration and data sharing than we see in other places. So we need to bring this data together to have a national wildlife database, because that will deliver robust monitoring, science, and management. And we need that for monitoring threatened species. We need it for making plans for management of threatened species and invasive species. We need it for high impact publications that are really going to change or maybe contribute to fundamental knowledge and important emerging knowledge in the space. We need it because our citizens like to go and see where their biodiversity is, right? So these are Atlas of Living Australia is a portal for anyone can go and look at what species have been detected nearby them. We also need it to measure our commitments for international organizations like the UN. We also need it in Australia because we have a higher than desired frequency of dramatic events. So we all remember the Black Summer bushfires. We were relatively flat-footed when this happened, meaning to understand the impacts of the bushfires on wildlife took us years, and we had very little baseline data to compare to. So we were not prepared to assess what was happening to our ecosystems when there was a dramatic event. But we also have disease outbreaks. This happens to show African swine fever, which is on the doorstep of Australia, but has not hit. But you could imagine the same type of graph for avian flu and other diseases. Climate change. We also want to be able to sell biodiversity credits along with our carbon credits. This means we need to quantify what levels of biodiversity we have before and after the program has been implemented. So who is using cameras in Australia? The answer is everybody. Not even that, just everybody. So what do we have right now for large scale biodiversity data, especially on terrestrial mammal, which is what cameras focus on? The Atlas of Living Australia, ALA, is our main data source. And this is really great and suitable for species distribution models, of which Australia is a leader, both in developing them and applying them. But these are essentially really good range maps. They're looking at the presence of the species, whereas camera trap surveys, because they have the absence, you can get more richer inferences. Those could include things like occupancy and abundance, and we can have more confidence in our results because we can account for biases associated with how the sampling occurred. And we often summarize all that as detection probability. We can account for detection probability. That's a really big advantage. We can also do the trends through time, and we can infer species interactions, both of which I'll show you examples of. So we also really have a lot of room to grow for Australian wildlife research into using a little bit more advanced methods to account for detection probability, because we know that this strongly can bias our inferences that we're making. So there's a lot of room for improvement there. What are the speed bumps to getting something like this off the ground? Getting a lot more cameras being deployed, being used, the images being sorted, that data being secured and shared. One of them is that there's just so much data. So each survey can produce 100,000 images. If you're taking video, it can be 50, 100 gigabytes. It can be a terabyte or more, depends on your settings. And we've already are aware of hundreds of studies occurring across Australia, of which the data is available for a dozen of those, less than 10%. There's an increase through time of sampling, but not matching what was predicted, which which was an exponential growth in the use of this technology. And what we think the reason is there's too much images, people can't get through it. So there's no point in setting more cameras if you can't go through the data you already had. I faced this before I had AI image identification software. And so this makes sense to me. But it's a shame because we can move toward an exponential growth, and I'll discuss how. And this all boils down to cameras are easy to use in the field, but generate unmanageably large data sets. So how do we solve those problems? So one, some basic things we can do is standardize deployments to make it really easy for people to set cameras. Have apps on your phone for setting them, automatically text the location of it and other information like date and time and brand, etc. And we also need secure storage for both the metadata and the images. Those should be done in the cloud so that people can access them anytime, anywhere. And then we need to be able to efficiently process lots of images into data sets. Okay, so we first want to pass all the images through a detector. This just assesses if there is an animal in the photo or if it's a vehicle, blank, or human. If it's an animal, it goes to a classifier. A classifier separates out which animals, which species it's most likely to be. And then we need to have all that data be passed through to some sort of national collation or synthesis center. And then we need to be able to let the people who provided the data access it, explore it, and we need to be able to share it with the entities that need it, like the federal government, threatened species or endangered species, EPPC Act. So the solution we came up with is the Wildlife Observatory of Australia. It exists, it's online, we're building the tools, some of them are already available. So this is a screenshot of the website. You can see our main products here. We're building a tech stack, completely free, limitless storage, infinite compute, and we will do almost everything for you. First is the image management platform. What we did for this is partnered with a European observatory called the European Observatory of Wildlife, and they had a platform. We collaborated with them to move that entire platform, copy and paste it all on Australian servers. The data never leaves Australia. It's secured, and the federal government and the Nectar research infrastructure will host all this. The second one is a tagged image repository for AI research to design better models that we can then take, put in our image management platform, so we get faster, easier identifications. Then we have a database where all the spreadsheet data is easily accessible, can be queried in a number of ways. For example, show me all the surveys that found spotted tail coal, or show me every survey from northern Queensland, period. Or show me every survey that has elevations at above 1,000 meters. These are all research questions that people are interested at JCU. So some of the key things that we're really aiming to achieve: more coordination and collaboration in sampling and fieldwork. This is essential for long-term and large-scale monitoring. As I explained, you can spend years out in the field and have 3 data points for tigers. Right? The same is true for all the species in Australia, and I'm going to give you lots of Australian examples in a second. We have now achieved this free limitless storage for all of your images, and can provide you spreadsheets for your analyses. We have options to make your data open access. You don't have to. I will say, just so people understand, that if you take federal funding or most state funding, you are technically obligated to make your data public. You actually don't own it. You have rights to it, but the government owns it. And I'd be surprised, even if you're an NGO, if you're not taking government data or government money, because that's most of the money. And that's what makes the research world go round. We're also doing training sessions for better statistical practices. And something that I'm really excited about, and we're getting pretty close to having done, is if you upload your images, not only can we get to a stage where you're going to produce a spreadsheet of all your detections for you, we're also going to give you like a 100-page PDF where we're going to have run every single species occupancy and abundance analysis with habitat associations on the supercomputers, and you just get a PDF output of every result you would want. This is super important for government employees on the ground out in a remote field station that are setting the cameras, and they want all their results, but they don't have time to do all those analyses, take the statistical training courses, don't have access to a supercomputer. There's so many things that we might have at a university, and we have the time to do it, and the postdocs that have the skills that people who are setting the cameras don't have. Okay, so some evidence to date. Lots of collaboration. So Bill's on this paper, 178 co-authors from Australia on one paper, talk about herding cats. And so this paper is about who is setting cameras, why they're setting cameras, what's working, what's not working, and how do we make a plan for the future. This is basically what motivated Wild Ops. We also were able to implement some of these things I was talking about for our largest single camera trapping study that was ever done in Australia, which is this WWF and Google's Eyes on Recovery after the Black Summer bushfires. Huge camera trapping effort, and we were able to have, they spent years not knowing what to do with this data. We were able to have everything processed and results to them for 93 different species in 8 weeks. Now we can do it in 1 or 2 weeks. Because the code is already there, and once the data is standardized in the right format, we literally just say, take these species, take that data set, run that same analysis, and give it back to you. So the scalability is in replicable code and really transparent, easy to use code. More evidence to date is our growth, which is exponential, unlike the amount of cameras being deployed. So we will have more than 1 million detections by 2026. Probably that's going to keep doubling. That's really great news. I will say a lot of these gray bars, those are blank images. So it just highlights the importance of having some computer magic help to get rid of all those blanks, get rid of all the easy to identify animals, and leave the experts to understanding whether or not it really was a potteroo or bandicoot. They're both facing backwards and looking the other way. That's where the human oversight really becomes important, not going through 5 million blank images. We also want to reduce this delay in when cameras are set to when we're getting the results about those things, so we can have management relevant timeframes for our outcomes. Okay, let's do some examples. So here's a map of data sets we currently have. The colors show which of the naturalized and invasive predators in Australia are present. So you got areas with just cats like Tasmania, areas with just dingoes in the center, areas with all three, and a long-standing debate in Australia that has led some of our most beloved scientists to never talk to each other again is the question, do dingoes suppress the abundance or behavior of foxes and cats? So this would be like a apex predator, mesopredator release question. So because this is so controversial, we went straight at it first. And we want to have all the different people on each side of the debate on the same paper with the same data and ask them, okay, if you don't like it, then you analyze it some other way and we'll all assess that too. Try to build this collaboration and coordination, right? And we also have a slightly different angle on it, which is, do these factors vary over ecological gradients? I have a particular hypothesis, which is that a dingo can kill a cat or chase a cat in a desert, no problem. But if there's a tree, the cat's gonna climb the tree, dingo's never gonna get it. So I predicted strong effects of dingoes on cats in deserts and no effects of dingoes on cats in forests. We got hundreds of different landscapes for each one of these habitats. Each of these landscapes had a number of studies in them. So what that means is we had habitats or combination of the species with not just 1, 2, or 3 samples, but entire camera trapping surveys, dozens or hundreds of times, really large data sets. The answers are dingoes definitely affect mesopredators in particular ways and in particular ecosystems. And there's no one size fits all answer. And partly that can help ameliorate the longstanding conflicts because everyone could be right for their study and their situation. So what this graph is showing is the green is the pattern of a dingo, over the course of the day with midnight in the middle. The blue is how the cat behaves when there's no dingo. Okay, it's a little nocturnal, but it's got some serious activity going on during the day and the crepuscular hours. And the red is the pattern when dingoes are present. You can see a pronounced shift toward nocturnality. Okay. Now, let's say the cat's favorite prey species lives in the day. This is going to potentially inhibit them from hunting that prey species, could create a temporal refugia for those prey species. And maybe it's a northern baycon, one of the local favorite threatened species. This result was only true in deserts. Oh, sorry, only true in forests, the opposite of what I had predicted. But we have a reason for that. All the animals are strictly nocturnal in the desert because it's so hot. They didn't have any room to change. They're just always not, everything's always nocturnal in the desert. So I was wrong. These are the results from a co-abundance model. What that means is we've analyzed both species abundances and corrected for detection probability and accounted for covariates like elevation, habitat integrity, and other things. And what you see overall is no significant trend. And if there was significant trend, or sorry, if there was anything, it'd be positive. Okay, so what that suggests is even though dingoes are displacing cats out of the temporal zone they would like to have it occupy if dingoes weren't there, that's not translating to a population level effect. So the cats are still there in what appears to be similar abundances. It's just they're a little more nocturnal. It's not that there's fewer cats. Okay, our future aim for Wild Obs is to contribute to threatened species index and reporting. So this is TSX, which we are in the process of being able to do that for them as well. This will add literally hundreds and hundreds of mammals and multiple time series. So now let's zoom in to areas around Cairns. So here are some images of cool animals right behind the university. So musky rat kangaroo, our most primitive animal. We got tree kangaroos, our most derived macropods. We have spotted tail coals, largest terrestrial predator on the mainland. And we have fighting paddy melon. So paddy melons are super cute. I bottle fed one yesterday, but they create a lot of images. So in this one camera trapping study, we had like 10% of all images were just paddy melons. And a lot of this is because you had paddy melons hanging out in front of cameras for extended periods of time. So again, a huge reason why we want AI to help us with this. So we also had cassowaries, which are, I think, one of the most iconic and interesting species that we have in the region. And since they're so big and I love megafauna, charismatic megafauna, we did a project on that. And this is led by one of the PhD students, Sky. And whenever you see the photos of people, these are their postdocs or PhD students in the lab that are leading this work. So Sky's PhD is on cassowaries. So she just got a paper accepted where she looked at cassowaries from camera traps across their entire range. What she found is most of the detections occurred within rainforest as opposed to wet sclerophyll or dry sclerophyll forest. This is pretty consistent with what we know about cassowaries. But within rainforest, they actually liked drier sites. We should take this with a grain of salt. Some of the wettest places on Earth are in the wet tropics of Australia. Drier is not dry. It's just not the wettest place on Earth. So when you take some of these, so when you have a model that has multiple covariates and detects and accounts for detection probability and use that to map the abundance of cassowaries across the range, you can see a pattern like this. Now, in this area, we're not predicted to see huge temperature changes from climate, but we are predicted to see rainfall changes. So results that link species to elevation and rainfall can be really important for predicting the outcomes from climate change. What we need now is better models about climate change and how it will exactly play out in these regions. Right, Alejandro? Yes. So some other things that were interesting is we got up to 26 different species on a single camera. So that is just like take a bow, Australian wet tropics. UNESCO is right. This is one of the most amazing places on Earth. But it wasn't all good. So here are some feral pigs. These we found were very abundant and widespread across the wet tropics. This image is a patty melon with her joey, and then a pig comes and chases them off. This is a cat carrying a native white-tailed rat. And so Zach had a question, which was, do invasive species suppress, or feral species suppress native species? And yeah, they get even bigger. There's some, yeah, cats here have amazing diversity of their body size. Okay, so what did he find? So this is using the co-abundance models again. And so this first graph is going to show changes in feral cat abundance and how that might affect whitetail rats. And indeed, we found a significant negative effect where as cats increase, one of their preferred prey species, this native rat, declines. So then to look at the other question based on that image we saw of a pig chasing away patty melons, we did not see a significant effect of pigs on patty melons. So that was two species pairs, a pair of pairs. Now let's look at every single species pair. So what we're going to do is if there's a significant negative effect, we're going to make that a red-orange bar. And if there's a significant positive effect, meaning they're positively correlated, we're going to call that a green bar. Then we're going to plot every single species pair where we had sufficient data. And we're going to make a histogram to look at whether or not overall predators are causing more top-down negative effects, these negative species interactions, or if they're generally green, means there's positive effects. In good sites, they're all doing well, and there's not a significant negative effect from the predator. So for cats, we actually had a fair amount of these red bars, suggesting cats are really causing top-down control in these ecosystems. For pigs, almost all of it was non-significant, even though we have lots of pictures of pigs causing havoc in these forests. The other animals don't seem to have an abundance shift when there are more pigs present. That's a good sign, and it was contrary to what we predicted. Okay, last slide where I review what we talked about. And since I promised to talk about global change biology, I'm going to relate specific examples to bigger topics. So when we talked about dingoes affecting cats and cats affecting mice, this is an example of how you can study trophic cascades. If you want to see other examples from how we did the same thing in Asia with tigers, you can look at some of the other work in the lab, especially from Zach. So deforestation, agriculture expansion, some of the two key issues globally. We saw how we can use camera trap data to look at how agriculture expansion influences some generalist species, and that can affect humans through crop rating and disease risk. We use cameras to look at how protected areas, how they're performing, and found variable results, great results for birds, complicated results for mammals. Mammals need larger parks, it appears. And we can even use cameras to start to investigate what are going to be the impacts of climate change. For example, that cassowary paper that you mentioned. With that, I'd like to thank the team at the Wildlife Observatory that's based at QSIF, Queensland Cyber Infrastructure Facility, but actually they changed the name to just QSIF, so it's just QSIF now. That's who runs Queensland's supercomputers, which I think JCU is a member of too. My lab are the ones that led a lot of that work that I presented. We have a lot of funders, especially ARDC. It's been a real big supporter. All the data contributors, both in Australia and especially from Asia, from the work I presented. And if you want to know more and get in touch with us, you can go to the website, wildlabs.org. If you want to know more about the research, it's on my website, ecologicalcascades.com. Thank you.
Okay. Questions?
Yeah, I have one online.
Oh. Noel, did you have a question?
Yeah, I do. Hi, thanks very much for that talk. It's really interesting, and I've been using camera traps for quite a few years now. And you're right in the analysis that it's really time consuming. Every time you put out a dozen camera traps, you come back with thousands of images, which take you days and days and days to go through. So that's a really beneficial thing. I had three questions, which maybe I should ask in sequence or um, give others a chance. The first one is, does it matter what type of camera it is? I'm using a Reconyx Hyperfire 550 white flash.
I'll start there. Yeah. We can include camera brand as a variable that can affect detection probability. Camera brand should never affect how many cassowaries there actually are, but it can definitely affect how many you detect. And so we include that as one of the many different things that can affect detection probability. Same as like how far is the field of view for each camera? That could affect detection probability. Now, does it matter when you're buying a camera? Yeah, because Reconyx Hyperfire is like $900, and the ones I use in my lab are like $100. The reason is, one, we don't have enough money, and two, a lot of ours get stolen. And so we just factor in that they're going to be stolen, and we don't worry about it too much. So if you have a place that you know your camera is really going to be intact and you plan to leave it there for maybe more than 3 or 4 months, I would suggest maybe one of these really expensive, nice and durable cameras like the Reconyx. If you're in a more sketchy area, which is like just any trail in Australia, people aren't even trying to steal them because they're being like thieves. They're just curious all the time. They don't even know. Or sometimes they want to use them to go hunting. They use them, people like them on the property. So basically those are the things I would factor in when making decisions about cameras.
Okay, thank you. Second is uploading images. How do you do that? Is it through a turn website process, or we're just got really slow upload speeds here, that's all.
Yeah, it'll be through a website not hosted by Turn, but hosted by QSIF, which will go directly to the federal government's infrastructure, which is called the Nectar Research Cloud, and is available to all universities and anyone setting cameras in Australia for free. The link to be able to do that is in the very final stages of being able to be ready to send out. And what you'll do is you'll log in with a simple email, and you'll click, I want to start a new project. It'll say upload a metadata sheet using this format, and then upload all of your images and folders where the folder name matches the camera name, and it will do the rest. And you could set that to upload overnight. You wake up in the morning and it's done, even if you got really slow internet up on the tablelands or on a remote research site. And also part of the reason we went with cloud infrastructure is Starlink and other satellite-based internet is already here. It's affordable. And so, we don't have internet because we're in a remote location. Was a really big concern, is no longer a concern. It's way cheaper to just get a month access to Starlink and upload everything than it is to buy a bunch of hard drives and store everything offline.
Okay, thank you.
And
the last question from me was, I noticed that most of your images don't show unbaited traps. Is that the preference for you?
Oh, no. Like, I didn't tell him to ask these. This is great. You asked the best questions. Baiting is something Australians do much more than in other places. There's a lot of pros and a lot of negatives to baiting. My lab doesn't bait because we use our cameras to study many different species. And if you're baiting cameras, you're going to highly shift the captures towards a subset of species. And that's fine if that's the only species you care about. But if you just leave a camera out unbaited, you'll probably get enough of the animal you wanted and many others. So we advise everybody not to bait. because you're probably gonna be able to answer your questions and help to answer others as well. But there's personal preferences for baiting and long-standing traditions of doing that. And again, just like camera brand, we just include type of bait as one of our detection covariates, because whether or not you bait does not affect the number of cassowaries there, just affects the number that you detected. And if you stick out fried chicken, which is the favorite bait for a lot of people around here and actually does work for cats, but if you stick out fried chicken, you might not get as many cassowaries because they're not cannibals.
It also changes the workload. You don't have to go out and bait every couple
Hey,
of days.
roadkill, I stand corrected. They will eat. Will they eat a dead bird? Okay. That was a bad example.
Thank you very much.
Great questions. Thank you, Noel.
Thank you, Matthew. Excellent talk. I'm curious about the Wildlife Observatory and a little bit of the wider vision of it. It is obvious, the example that you have show the potential for scaling up. So we have individual studies that, or individual questions that can be answered with the small studies, but once you scale up, you can answer bigger questions. But my question here is, what do you think could be other potential applications of this in terms of answering new or maybe old hypotheses that we never had the data for, or developing new concept, or advancing ecological theory or modeling in other aspects that weren't possible before just because we didn't have access to such data? Is that something that you're currently exploring, or you have some ideas of new things that can be done rather than just scaling up?
Great question, Alejandro. Yeah, there's a lot of research frontiers that I think cameras can be used for, and we have not been able to use cameras for a lot of these that I'll talk about until recently because they're so data hungry. I'm referring specifically to structural equation modeling and food web modeling, where you have many species in a single model and they're all interacting. And so there's also a lot of noise. And so to be able to disentangle detection probability from interactions with habitat and interactions with 5 other species requires so much data. And I mean, really like hundreds of thousands to millions of trap nights to be able to confidently say anything. And so I would say the frontier is understanding entire food webs and what the effect of picking out and putting back a particular species. And I know that sounds like, oh, we'll never be able to do that, but like, it's very likely all the pigs are going to be gone for 1 or 2 years when African swine fever gets here. Like, we just saw this exact thing happen in Asia, and it was fascinating from a research perspective and horrible from an ethical perspective. But like, that gives us this opportunity to have these natural experiments. You know, bird flu is on our doorstep. There's other diseases that pop up and go away. There's big efforts to cull certain animals, or just like get all the cattle out of the rainforest. Okay, so that's going to have a huge effect overnight if people actually put a lot of effort into doing that. We can predict how that will affect not just the one animal that instigated this conservation intervention, but how it could cascade and affect all the others. The future beyond that, probably looking at diets, so assessing how these food web effects also cascade to the plant, which is really important. A lot of our plants are either pollinated or dispersed by animals, or there's predators of these seeds. So they're eating the seeds or eating the seedlings. There's a lot of food web analysis that can be done. I foresee structural equation modeling being a frontier there. There's also Bayesian network models. And the limitation to date has been data. And so the big data revolution that cameras are a part of should be able to address that. That's what I'm excited about.
Other questions? Yeah.
With your researcher in the wet tropics area, did they find that there was any increase in the dingo numbers, especially since Cyclone Jasper?
Such a good question. So we have not done any temporal analyses to date, except for time since fire for the Black Summer fire study that I mentioned. So one of the goals for the Wildlife Observatory is to have replicated surveys through time, but to track mammals and other animals that get detected on cameras using this robust metric. We have not done that yet because we don't have enough time. Like, that's the next thing on our list, and we're really excited to do it. Specific hypotheses like how does a cyclone affect 1, 2, whatever, the whole community? That's a really interesting question. Would love to come back in a few years and give you the answer. Or if you have a postdoc that wants to do it.
I might be able to give you a bit of anecdotal evidence here. We're with the Rainforest Conservation Foundation. We're on the Cooper Creek. We have 3 properties, 19, 13, 9 hectare. We have a cachery that lives on the property, been there for 19 years. He generally has 2 to 4 chicks, has successfully raised and sent off over 40 chicks. The others have died, generally through the idiot locals who consider a 60k zone to be 100. But since Cyclone Jasper, from the ferry to Cape Trib, there were probably about 156 landslips along the McAllister Range, some small, some large, and they have actually become highways. Now, we have always had pig traps on the properties, being managed by the council, and the pig population has been manageable. Very rarely would you see a dingo, perhaps one this year and one 2 years later. Since Jasper, the pig population has exponentially increased to the stage where most of the properties up there have to get shooters in, quite often getting 7 a night. The property across the road has cameras because they have commercial walks. They're picking up 20 in a pack. But we've never had a dingo problem.
Dingoes are a pack of pigs.
Pigs. Yeah, now with the dingoes though, we've never had a dingo problem, and they're now actually, because some are small and some are huge. Some of the dingoes are a colossal size. They're crossing with pig dogs, and they're actually pulling down the cacheries, which a normal smaller dingo won't. But once they cross with a pig dog, and we've lost all of our 4 cacheries this year, we think, through the dingoes, which has never been the case.
Well, we need to get 100 cameras out on your property forever, starting in 1 month when Sky, the student studying cassowaries, is going to be here studying 400 cameras. We spot another 120. So let's whatever, all the things you're saying, I think are so fascinating. And I think cameras can be one of the methods to give you some insights. Another is going to be, whenever you see a dingo poop, pick it up and put it in the freezer. I don't have a student do anything right now, but when we do, we're going to empty your freezer and see if you're right that they are eating cassowaries, because those are some pretty big accusations you're making. And I think they're probably right, but we can know if we get the cassowary down. Oh, sorry, if we get the cassowaries in the dingo down.
Oh, you got one? Sorry. We have one online. Yeah, question online? He can't talk? Oh, okay. So I'll come back to the microphone there.
Okay. This is from Patrick. I'm interested in how often false positives stroke negatives on mistaken AI detection influence your results. Are there consistent ways to statistically account for biases like that?
Great question. So currently, we still have a human verify all the images. That is really easy when you're given a set of thumbnails, let's say a 5 by 5, and in the computer vision says, I think these are all dingoes. And you can just look through a bunch of pictures and be like, that dingo is a collar, it's not a dingo, that's a domestic dog. Or, okay, I can see that one kind of looks like a dingo because it's run away. That actually looks like the tail of a different animal. So your eye, you can do 25 every like 3 seconds of verification. So it's already a huge win. But once we get to a level where we assess the accuracy and precision of the AI is over 98%, which we're getting close to on a lot of species, humans are not 100% either. And so the false positives become not important at all for 95% of species. but could still be important for the rare species. So let's say we got night parrots, almost never detected, very small range, very threatened. Every single positive detection of a night parrot should be reviewed, because if it really is a night parrot, that matters. And also the training data for night parrots is going to be very sparse. And so the model will likely never have a really high accuracy and precision and recall for that species. So the way I think we're going to deal with it is for common species that have really high accuracy and precision and recall, we're probably going to eventually get to the point where we'll never have to look at them unless we want to. And for rare species, we'll always review them because the models will probably never have enough training data to get a really good estimate. And either way, we want to just double check, and people want us to picture night parrots. So I kind of talked around that. And I think a bigger way of saying this is we're figuring that out now and trying different things, but I suspect we're going to move further and further into a place where we just trust the AI.
I can just give a bit more on the dingoes there. I hate guns, but I went up there to see what they do and what happens. And they normally, they're up there for pig shooting, of course, but our conservation officer for a week puts out bananas and coconuts in the same spot, so the pigs know they're there. And so they turn the spotlight on. Here was a dingo with a banana in its mouth. And this thing, definitely a dingo shape, but I kid you not, it was almost the size of a small donkey. It was huge. Yes. And the last chick, Cassowary chick that went, the male came in the next day and he was totally distressed, feathers missing from the front, which makes me think he was part of the attack or defense or whatever. And the pigs can't do that. It has to be a dingo.
I would just will say pigs do hunt, even mammals, small mammals, but that I don't know the frequency, but they observe hunting, especially lambs. They're known for hunting lambs. They would definitely be capable of hunting a cassowary chick. I'm not saying that they did or did not. So we really need to get into the diets of these dingoes to see if there's banana or cassowary and the frequency of these things. There's some canids are omnivorous to different degrees. So, you know, my dog will eat anything. So.
Did you get, sorry, I'll just, did you get any evidence that dingoes are affecting pigs?
Zach is the person who would know the answer to that. Zach, do you want to hop off audio on Zoom and answer?
I don't actually know off the top of my head. I'd have to dig back into the results, but I don't think it was a significant result, but I think that also might be an issue of overdispersion in detections. Both two critters with large group sizes, hard to model. So still exploring that one.
And I can explain overdispersion, which is a serious issue when you're dealing with these data sets, because you know, you assume some sort of distribution in almost all statistics. And cameras and many wildlife sampling approaches, not just cameras, but cameras, we have so much data and we have higher quality data. We get what's called zero inflation. So, you know, you're going to have 50 days with zeros. Then you get to pack 20 dingoes. And so the zeros are zero inflation. And then, you know, at the other cameras, you get 1 or 2 dingoes every single day, right? And, you know, you get a normal distribution around 1 or 2 individuals every 2 or 3 days. And then you have these 20s way out on the far end. And so dealing with zero inflation and overdispersion was one of the main topics of Zach's PhD. And we are getting to a place where we're a lot more confident in making and addressing and making statistics that make sense, even if the data looks messy. But it's a very big issue that comes out strongest when you have 2 species that are prone to a bunch of zeros and then huge group sizes for like 20 or 30 individuals, especially because we focus on a type of model called an N mixture model, which unlike occupancy models, which is just presence or absence, or a Royal Nichols model, which is an abundance model, but just as the frequency of detection, so there's no group size in it. We like a model, like an admixture model, that includes group size as one of the parameters, because one dingo in a pack is very different than 5 dingoes in the pack, which is very different than 10 dingoes in the pack. And we like to capture that information in our analyses. The problem with that is then we get over dispersion. So we're cutting a fine line, and it's the frontier of statistics in our field on this topic, and it's not settled. But dingoes and pigs would be the worst case scenario for these kind of models. Yeah.
One or two more questions.
Yeah, I've got 2 questions, and prefacing them with an apology. I'm an engineer and know absolutely nothing about ecology, so if they are slightly naive, forgive me. First question is, you're using cameras to trap and observe. Colleagues here are using acoustics to observe. I would assume acoustics to produce significant data sets. What are the benefits of a fusion of acoustic data with camera trapping data, number 1? And number 2, what would that require infrastructurally? So you've had to build a whole infrastructure around the management of the data sets you're collecting. And number 2, is the community thinking about these sorts of integrations? So that's the first question. And the second question, somewhat more self-serving from an engineering perspective, is in terms of your technological infrastructure for observation, are you living in the best of all possible worlds already, or are there technological developments that you wish you had as an ecologist that require research that go beyond making a device faster, more water resistant, having a longer battery life? Yeah, I was just wondering whether you had some comments on that.
This is the best set of questions from a group I've ever gotten. So great questions, guys. So the ecoacoustics group in Australia is called Open Ecoacoustics. The person who is in charge of that is named Paul Rowe. He just moved to JCU. He's down in Taxville. And one of the main collaborators is Lynn Schwartzman, who is online, I believe. Sorry, Lynn. And so there's an investment from the same organization, ARDC, Australian Data Research Commons. They have a planet initiative, and they want machine-based observations of the planet. And so they have cameras and acoustics are the 2 main projects right now. We work, I would say, hand in hand, but not on the same exact platform, but talking to each other constantly to achieve what you're talking about, which is one day, most of the animals that make a lot of noise are hard to detect on cameras. Frogs, birds, okay, these are vocal animals that don't show up on cameras very often. Whereas mammals tend to make less noise, so don't show up on acoustics and are better for cameras. So there's complementarity that's really beautiful. And in the future, we'll be able to monitor our entire community using a single device that records sound and video. And then we just run an AI and delete everything without a good sound or a image that's not a blank image. So that's the future I see there. Yes, they will be fused and it'll be wonderful. The other one is, what is the technology around cameras that needs to be developed in the field? Okay, I think it exists, which is the biggest cost now is not the camera. It's the PhD student to go out there and set the camera. And for the government, it's the salary of the state ranger who's setting those cameras. And for the landowner, it's getting out there himself to go and set, collect, check. And so with the Internet of Things and getting let's just say signal further and further, you could imagine setting cameras once and having a solar panel and a connection. And so all the images are transmitted back to you and you never have to go back for years on end. That exists already in places that are sunny and have good internet. That does not include anything within 100 kilometers of JCU, but it could include everything within 100 kilometers of Perth already. So I would say, yeah, connectivity. So to reduce our cost of setting the cameras.
Last question, please.
Okay, so it's on the cassowary data you showed. I actually think cassowaries really like the very wettest forests, but when you see they're most active in the dry season when there's no fruit. So that's when their activities really increase. So dry season activity really increases, and they tend to get really concentrated in the swamps where the palms are still fruiting. But if you see where your map was, you had them on the top of the mountains and Daintree, and they are the wettest forests in the wet tropics. But they're really active in the dry season because there's no food.
Okay, do you want to be on Sky's PhD?
Well, there's probably better people than me, but I just had to think about why that was, but now I figured it out, yeah.
Okay, so when Sky comes up here in a few months, I'm gonna make sure she visits and, shares what she's doing, and also has long conversations, and maybe you can advise her on methods to increase her knowledge. So long-term data sets, like throughout an entire year, instead of 2 month at 2 month. So instead of doing a 2-month single survey and running away, which is what we often do, leaving them out all year could be useful to look at these seasonal habitat use patterns. That's really cool. Yeah. Thank you. Thanks. Thank you.